Thanks for joining this presentation. It's an unusual year for PEGs, but I'm glad you're able to join us today to talk about de-risking antibody lead selection. We subtitled this presentation, "Is your antibody as specific as you think?" And the reason, of course, we did that is probably not. There's a good chance. In our tests so far, we found that about 25% of antibodies that we've tested are polyspecific. That is, they bind not just the target protein that people think they're binding, but also another target protein. And that's a significantly large amount of off-target binding, much more than we expected. That's, of course, significant because off-target binding is the single largest cause of preclinical failure, at least in the studies that have been done to date. A study by AstraZeneca a number of years ago found that off-target binding was the single largest cause of preclinical failure, 62%. Actually, it was the single largest cause of early clinical failure as well, even after preclinical. Off-target safety was the biggest predictor. And that was for small molecules. We haven't seen comparable studies for antibodies yet. But what we know from our tests and from others is that somewhere around 25% of antibodies just target something else as well. And if you don't know that, you're taking risks for your antibody in clinical trials as well as preclinical trials. And I should mention that polyspecificity that we've seen is not to related family members. Those have already been tested usually before they get to us, and by the time certainly they get to preclinical studies. We're talking about polyspecificity binding to completely unrelated targets. So think proteins that are really very difficult to predict. So at Integral Molecular, we've developed a number of platforms against membrane proteins to predict specificity and to develop therapeutically relevant and developable antibodies. We've been doing this for 20 years. We've worked with thousands of different membrane proteins. We have an entire antibody platform devoted to discovering highly specific, highly therapeutic functional antibodies against GPCRs, transporters, ion channels, viral envelope proteins, immuno-oncology targets, and so on. We've developed a number of platforms that are used by over 300 companies. Every major pharmaceutical company has used our technologies. They've been published in over 250 papers and patents, including papers in Cell, Science, and Nature over the years. And we're still very active, collaborating and partnering with different companies using our different platforms. We have a number of different platforms. I won't go into all of them today, but we have platforms in target discovery, target engineering, antibody characterization engineering. Many people are familiar with our lipoparticle technology for antibody discovery. And I'll talk today mostly about shotgun mutagenesis and the membrane, a little bit on that, and the membrane proteome array, which we use for specificity testing. So the membrane proteome array is a collection of essentially all or nearly all of the human membrane proteins in the human proteome. So we've collected about 6,000 human membrane proteins. That's about 5,300 unique membrane proteins, almost the entire human membrane proteome, 94%. And we've expressed each of them in live, unfixed human cells. That's important, the unfixed part, because the body's not fixed. Therapeutics do not see fixed cells. And if you want to test for specificity, you have to maintain the native conformation of the targets. So all of our studies are always done in unfixed cells. And as you'll see, many are done in live cells. We do this in 3D 4-well plates. So we detect by flow cytometry, the most sensitive method out there, to detect off-target binding as well as target binding. And at the end of the day, once we've put an antibody or other therapeutic across the arrays, measure binding, we deliver a profile of the reactivity of that antibody against the human membrane proteome, usually just the target, but in many cases, we see the off-targets binding as well. We think this is really important because what we've seen over the years and what we've been doing lately is characterizing antibodies that are in the clinic and even approved on the market. And we see, as I said, almost a quarter of antibodies seeing things that they're not expected to see. So these are some examples that certainly scare us, and if they're from your company, would scare you. These are antibodies, and this one on the left is from a clinical stage. It's in the clinic right now. It sees the target, but it also sees an off-target protein. This company is developing this, is actually purchased for over $1 billion. And this off-target, it kind of looks low here, but it's actually highly expressed throughout the body. The antibody is intended for cancer, so cell killing, which means that any cells that express the target or the off-target are likely going to get killed. So not a great predictor for safety profile. This antibody actually went through that whole process, the clinical process, and then got pulled because it killed patients in clinical trials. It hit the target, similar to this other one. It also hit this off-target, which was actually an immune checkpoint protein. And we believe because it hit that off-target, it ended in patient deaths, and that antibody got pulled from the clinic. What's possibly even more disturbing is that we're seeing antibodies that are actually already on the market, that are FDA approved, and are seeing off-target proteins, many of which have severe safety issues. So this antibody on the left is an FDA-approved antibody. It sells $500 million a year. It hits the target, but it also hits 2 off-target proteins. I should mention these 2 off-target proteins are actually family members. They're closely related. So it's not that surprising it sees both of them. This antibody is in trials for additional indications, and we'll see what happens with it in terms of safety. This antibody already has black box warnings. It's an antibody with a $20 billion market. It's growing, and it hits it off target pretty significantly. We think that this off-target binding is causing those black box warnings, that the safety profile is directly related to the off-target binding. And we're in the process of examining many of these antibodies right now to make that link, to make that correlation, and publish those results. Of course, waiting to the end till you get to market or until you get to clinical trials is not ideal for specificity testing. So what we recommend, of course, to all of our customers, whether you do it through us or anyone, it's really important, test early. We think that early lead selection testing is a great stage to test. You've got alternatives if something hits an off-target, other backups that you can still turn to without canceling the entire program. So this is an example of one project that we did with a partner who gave us 2 antibodies in this lead selection stage. Both antibodies hit the targets in blue, but one of the antibodies also hit an off-target protein in orange here on the array. And what was more interesting is when they put these antibodies into mice, they both went to the thyroid, where the antibody is actually supposed to go, where the target's expressed, but this antibody also ended up at the adrenal glands, where the off-target protein is expressed, meaning that our specificity screening predicted the off-target binding in vivo, and the safety issues that would have involved if this antibody had progressed. Obviously, they ended up going with Mab number 1 into IND filing. Our platform has been tested with just about every biotherapeutic type at this point. Certainly IgGs, monoclonal antibodies, polyclonal antibodies, bispecific antibodies, CAR T cells, whole cells, functionally as well as in binding, SCFBs, of course, peptides, nanobodies, pretty much every biotherapeutic modality you can imagine we've put on the array. We've optimized conditions, and we've been able to test for specificity as well as deorphaning when you don't know what the antibody target is. So from that perspective, what we've done, some examples, in this case, we took an orphan antibody. We knew it hit a cell type, a protein on a cell. We put it on the array, and we were able to determine that it hits GLU8, which is a glucose transporter, or 12 transmembrane transporter that doesn't have any antibodies actually before that. We're also able to do a number of different proteins. This is one of the proteins we've done. We've done many, many others since then, more for target discovery, for new targets in immuno-oncology and Alzheimer's and metabolic diseases and viruses. But in this case, we took PD-L1, we put it on the array. We identified, of course, PD-1 as well as CD80, which are known ligands for PD-L1. We've done this for other proteins where the protein was the partner protein was not known. It was orphan protein, and identified partner proteins, deorphaned those proteins to establish pathways that those proteins use and new targets that people can go after for immuno-oncology and other diseases. At this point, we've actually done this hundreds of times for specificity testing, as well as even just for deorphaning. We've done this hundreds of times to identify the targets for unknown proteins, unknown antibodies. Our platform is the largest array available out there. It's also the most versatile. So we have 6,000 proteins in our array, 5,300 unique proteins, which is 94% of the entire membrane proteome, more than any other company has. We do everything under native, unfixed conditions. That's our standard binding condition. We do for every project. Our membrane protein library has been completely optimized. We've been working on membrane proteins for the past 20 years. We've worked with thousands of different membrane proteins even before we launched the Membrane Proteome Array. They're all tagged. We know they're expressed. Many of those plasmids have been optimized for expression. As many people know, membrane proteins often don't express all that well or traffic all that well, so many of our membrane proteins have been optimized for those. We use flow cytometry for detection, which is the highest sensitivity, most quantitative detection method out there. As part of what we do as a standard process, we do a FACS dilution curve on all hits that come out of our libraries. That's just part of the process. We do that every time for every customer and every project. And we do all that in 4 weeks, faster than anyone else can offer. I'll also touch on a couple things that we do that are kind of unique that don't come up as often, but come up frequently enough that demonstrate some of the versatility that we have with our platform. So in many cases, for instance, we screen on HEC293 cells. They're human, they transfect easily, they're easy to work with, and they're well used throughout industry. But of course, HEC293 cells express many membrane proteins on the surface of their cell endogenously. And in some cases, those antibodies or those proteins bind to HEC cells even without any other exogenous proteins being expressed. This is an example of a commercial B7H3 antibody that we tested on HEC293 cells. And it's buried in here in the noise, but it basically didn't give any background. It's because the antibody bound to everything in the library. The whole library here was expressed on HEC293 cells, so you can't differentiate a signal from anything. We took that same antibody and we tested it on avian cell line called QT6, and we're able to eliminate the background binding and clearly differentiate the binding to the actual target here. I should mention, we screen, for every project, we screen human HEC293 cells as well as quail cells, the QT6 cells, in our assay setup to identify the best cell type for that particular antibody. We don't want to go into a screen of the entire array and get background like this. We do that so that therefore we screen which cell type and which concentration of antibody. We do all of that up front, and then we get very nice screens on the back end. We also, of course, use live cells, and that's really important for native binding, native conformation of all of the membrane proteins, but it also allows us to do some unique things with our library that no one else can do. So for instance, we've been screening viruses for infection of the library to identify new receptors for many years. This here on the left shown is dengue, where we put a dengue virus on the array, and we've identified new receptors shown in orange. that allow the virus to get into cells, as well as known receptors in blue. You'd expect to identify those, and we have, but also new novel receptors. We've also done this for Zika, for Ebola, we're doing it for SARS, of course, and several other viruses to identify novel receptors. And I should say, in every case that we've done this, we've identified new receptors or new attachment factors that allow the virus to get into otherwise non-permissive cells. In addition to viruses, we've also done this for cells, live cells, putting live T cells in this case onto our array and looking for other membrane proteins that activate those T cells. In this case, it's CD69 overexpression. In this case, we're able to identify new immuno-oncology targets, some that are expected, like CD86, and some that are known, like in blue, but about a dozen other membrane proteins that were novel, that have never been identified before, that activate these cells. We did part of this as a collaboration with Daiichi Sankyo. A successful collaboration is now completed that has enabled them to identify new immuno-oncology targets that are going forward in their discovery. I'll also touch on some interesting examples of why antibodies see off targets. This is something we've been able to dive deep into using some of our other technologies. So a couple of years ago, we published a paper discovering antibodies against GLUT4, a 12 transmembrane transporter. It's also called SLC2A4. We discovered a number of different antibodies against GLUT4. GLUT4 doesn't have any other antibodies against it, any monoclonal antibodies that can do FACS and see the native extracellular surface. So it was a very successful program from the antibody discovery side. But what was interesting for this particular talk is that we discovered some antibodies that also had a small amount of off-target reactivity. And again, that's not unusual. We had many antibodies that were specific to SLC2A4, but about 25% that saw something else. One of those something else's was Notch1. You can see this antibody here bound SLC2A4 in blue, but also saw Notch1 as an off-target. And the interesting thing about that was that Notch1 and SLC2A4 have no sequence identity, less than 7%. They're not even in the same class of family. GLUT4 is a 12 transmembrane transporter. Notch1 is a single transmembrane signaling domain. So why would this antibody possibly see both targets? And we were able to answer that question. That's kind of buried in this paper, but we were able to answer that question as part of our epitope mapping. So we have another platform called shotgun mutagenesis epitope mapping. Won't go into all the details, but we make comprehensive alanine scans across a protein. We can test all of them by flow cytometry. We've done 1,000 of these to date, mostly on complex proteins like GPCRs and transporters, ion channels, viral envelope proteins. We have a 95% success rate, even for very difficult antibodies like conformational antibodies, very difficult proteins like GPCRs and transporters. And using this technology, we're able to epitope map exactly where that antibody bound. So what we're able to determine was that that particular antibody bound to this loop, in particular these residues, leucine, glycine, and proline, residues 61 through 66. Saw this motif. And that motif is actually constrained by the transmembrane domain. So it forms a loop, an extracellular loop that you can kind of see here. What was interesting is that when we look at Notch1, we found the exact same motif, also in a constrained loop. So this is a portion of the protein, but it's the portion that we believe the antibody binds. It has the exact same sequence motif, LG, XX, GP, same spacing, same motif. It's also contained within a loop. In this case, the loop is constrained by a disulfide bond that holds the ends together, but forms a very comparable binding epitope. So epitope mimicry across these proteins, even though these proteins have no sequence identity, less than 7%, you'd never be able to predict this cross-reactivity unless you started epitope mapping at the amino acid level to uncover exactly how the antibody is binding these two proteins. So more of that story is published in this PNAS paper if anyone's interested, but explains why we're seeing the kind of cross-reactivities that we're seeing, the kind of polyspecificity that we're seeing across many different antibodies. All the things I've talked about today, our epitope mapping, our membrane proteome array, are all available on a fee-for-service basis. We do this all the time, literally hundreds of different projects available through scientist.com as well as the Science Exchange. You can call us directly and we do this. We also sell lipoparticles as a reagent, and then we do a number of partnerships for new target discovery, for instance, for some of the immuno-oncology targets or viral targets, and some of the antibodies that we develop are more on the licensing side. Thank you for joining us. If you're interested in any more of the antibody discovery, my colleague Ross, is going to be talking about antibody discovery tomorrow on September 2nd. Actually giving more of a tutorial about how our best practices, the things that we've learned about how to discover antibodies the best way against multi-pass membrane proteins. At this virtual PEGS, we've also got a number of virtual posters from Ross, David Tucker, myself, and Sharon Willis on different technologies. And we also have a virtual booth. Come visit us in cyberspace, I suppose, and we'll see how that works. But we're here, we're available to you. If you have any questions, we're always happy to answer them. My email's there. If you can't find us anywhere else, feel free to email me. Thanks so much, and enjoy the rest of the conference.